Kia ora. Welcome to Lincoln University and to our flagship building, Waimarie. Welcome to the Excellence Series. And before we get into it, and I will introduce to you Professor Grant Edwards, our Vice Chancellor, and he will take care of the rest of the day introducing Amanda. Just want to do some housekeeping. You already heard, some of you probably already heard the fire alarm. That was a test at 4 o'clock. I don't expect any more tests. So if we hear it again, it is the real deal. And probably the easiest thing that we do is take the doors that are behind us. Unless there is danger, then we'll take those doors and you can follow us and we'll make sure that we keep you safe. Toilets are, if you go out this door, they're actually just across from this room. After the presentation, there is a chance for networking. We'll have some beverages and something to eat. We will be serving alcoholic beverages. So I would want to let you know that your family, your partners, your whānau that are sitting back home wants you home safe. So therefore, please consume responsibly. So without that, I'd like to introduce to you Professor Grant Edwards, the Vice Chancellor of Lincoln University.
Sure. Thank you, Ben. Ena mana, ena reo, ena hu, tēnā koutou, tēnā koutou, tēnā koutou katoa. It's a real pleasure to have you all here today to continue our Excellence Series. The Excellence Series at Lincoln University is about how we can contribute deeply to the public debate on many of the critical issues facing our country and our communities, while showcasing our world-leading research and education for the land-based industries. It is a real pleasure tonight that we're going to hear from Professor Amanda Black, as she talks about how our fragmented landscapes and multiple disturbances have impacted on our Karri forest's ability to thrive in the 21st century. A little bit about Amanda. Amanda is the Director of Bioprotection Aotearoa, a Centre for Research Excellence, which is hosted here at Lincoln University. She has a very strong background in Mātauranga Māori and the ability to facilitate teams of people and bring together excellent collaborations. She is a Rutherford Discovery Fellowship recipient and has held a number of ministerial appointments within the Department of Conservation, Environment, Education, and the Ministry for Primary Industries. Amanda's research area is in environmental soil chemistry and biochemistry, focusing on the health of soil ecosystems. Her current areas of focus are an understanding of the relationship between gene expression and soil productivity, such as carbon storage, halting Karri dieback through the discovery of disease-suppressive traits in soils, and focusing on biosecurity and climate change that are of concern to Māori and Indigenous communities. No mai, Amanda, welcome.
Thank you, Grant. You pretty much gave about half my talk there, so maybe I'll just let you continue. But it's thank you, Vim, for MSC and Katie for organising this. These things always take more effort than people realise, so just putting the thanks out there, and I know that you come from a difficult situation, just been one of those years, huh? It's really good to see a lot of my friends and colleagues here, and some people I don't recognise, but that's great too. When we talked about this being sort of a professorial talk, I had visions of having to wear a gown. I'm so pleased that didn't happen, and I have to sit there with my books, go, learned colleagues and friends. So I don't have to do that either, so I can just kind of be me and talk you through this research, which has been a passion of mine for the last 7 years. I've kind of done it many times on the smell of an oily rag, and through this journey, I like to think I've had impact, but I've met some really, really great people along the way. And hopefully some of that passion and the findings that we've come through in the last 7 years, we can highlight in this presentation. But I'd also like to thank the Rutherford Discovery Fellowships for funding at least a third of this, the Marsden Fund, and also the Tertiary Education Commission from where this all started. So thank you. I should do mihi tū, so sorry for getting here. Mihi tū to Taumatu mana whenua here for the manaakitanga and allowing us to have this building on their land. So, as Grant said, this talk is about my interest and the research and the people that have joined me on this journey, talking about our unique coated forest. And when I say unique, so when New Zealand parted ways with Australia geological eras ago, it came with a unique set of flora and fauna, and over time they've become more unique and very vulnerable. And specifically, I'm talking about our coated forest here, and the natural, this is a laser pointer?
Yeah,
Right.
That's great.
I don't know what screen to look at, it's terrible. So the yellow, I won't do it because that's kind of confusing. The yellow line represents where coated forests now naturally occur. Now they parted ways when we took off from Gondwana, and they came along with us, and their lineage goes back to the Jurassic, which is a very long time ago. I won't embarrass myself by trying to remember that kind of era. And they now are a remnant of 21 species around the Pacific, and they're part of the Agathis family. So the New Zealand endemic coated is Agathis australis, one of the 21 species, just really remnants around the Pacific now. And it's really a subtropical species that's confined to that sort of Auckland level and above. Used to cover 1 million hectares, but due to our land use changes and deforestation, they're now really around about less than 60,000 hectares of what we have. But not only are they a really long lineage, historic geological lineage, they also long-lived trees. So they live to around 1,500, 2,000 years. And because of this, they tend to drive the ecosystems around them. So they might not be numerous in the forests, like this picture from the Kaipara Harbour in the early 1800s depicts. Like, that's how close they came up to the shore. But their size and the age lets them drive the. So my whole team have just come in late. And I've pulled them out. Sorry. Because of that, they drive the ecosystems around them, including the codependency and co-evolution of many, many plant species that we do see in these forests, native orchids, epiphytes, ferns, all sorts of things, invertebrates, and some of the soil formation. So some of the I guess the soil microbiome will be unique and indigenous as well. We'll go on to talk about that as well. But the soil formation that happens under these trees are also very rare for soil scientists in the room. Podzols, they have this unique profile here, and that's because of the massive amounts of leaf litter and tree bark that falls around them, causing this leaching effect that you see in this impressive soil profile here. So this loss really of an iconic species that's happened really since the 1860s. We've reduced these forests to about 60,000 hectares, and about 4% of that is in highly modified small remnant fragmented stands, and of this less than 1% is actually original old growth, so ancient forests. So those really, really big trees, that the ones we see on postcards, the ones that you sit through endless amounts of in New Zealand safety videos that you have to watch again, but these are the ones that we're talking about. And you know, so Kauri were cleared for a major source of timber. A lot of it went over to British the boat industry, and also to rebuild San Francisco after the earthquake. And after that, the resin was dug up through gums, and we had land clearance for agriculture. And in the 1950s, when New Zealand Forestry decided to expand and experiment with species, we had these introduction of plant pathogens through that plant trade nursery movement that has become the issue that I'll talk about today. In the 1950s, there were two sites. We call them Ground Zero. So there was Waipua Forest, which is part of this, the research that we've been looking at. So Waipua Forest is home of Tāne Mahuta, the big picturesque kauri that we often see on the TV and the ads. And there was a site for where the New Zealand Forestry would decide to start a nursery gathered from kauri around the Pacific. I talked about those 21 kauri species. So we had kauri come round from Australia, from New Caledonia, and from Fiji. Different specimens there, and they started up nursery and forestry there. And they also did that on Aotearoa Great Barrier as well. So that's another site we call Ground Zero. And this, the map that you see on the left, right, the one, whatever I'm going to get wrong. Anyway, so that's the latest surveillance map from MPI that shows the distribution of Coyote dieback that we now see. So pretty much it is everywhere apart from a few pockets. Now that was because we didn't have those molecular diagnostic techniques back in the 1950s. It was misclassified as a Phytophthora, which is those people plant pathogens in the room. Those not. Phytophthora is a plant pathogen cause for the potato famine, the Irish famine. So that was a Phytophthora species. It's Greek for plant destroyer. They observed lesions in the trees in these two forest areas in the 1950s, called it Phytophthora heviae, but it was only until 2015 when the signs were getting worse and more trees were getting affected, and we had molecular diagnostic techniques that it was actually finally named as Phytophthora eclodicida by Ben Weir, and it was new to science. If those interested, belongs to a subtropical species of Phytophthora. This is a soil-borne pathogen, so it comes in through damaged roots and affects the tree. When you see the crown canopy, it means the tree is sort of dying from the canopy down. So effectively this comes in and it starves the tree from the top down. The zoospores that we've got a video on the side there, that's from Wanaka Girth's lab. So that's the kind of part of the life cycle that's going to seek out the roots and infect the trees. And they have a nifty way of snipping out their host through chemotaxis. The life cycle of Phytophthora, and it's not a fungi, but it's fungi-like, is complicated, which means the tools we develop to try and control this are complicated. It has 6 stages, a life cycle there. So the key ones to look at here are the oospores, which are those the survival structures. They hang around in the soil for years until the conditions are right. They produce these things, sporangia, which are fruiting bodies, and from that the zoospores, which are the things you saw in the video, and those are what seek out the hosts to infect the trees. There is no known cure, there is no effective treatment, and it seems to have 100% mortality in the trees that it finally infects, and that goes from the seedling right through to those mature trees. So the rest of the talk that I'll go on to describe is really the research program that I've developed over the years with multiple funding streams and multiple people and a lot of great students and postdocs. The first was what we'll talk about is the land use effects on pathogen growth and virulence. This led to looking at the impacts of soil microbial community and soil function from those land use changes, as well as the presence of the disease. I'll talk a little bit about the Indigenous knowledge guided management and our search for the biological control agents, and onto the new research, which is recreating those resilient coyote ecosystems, which is part of my Rutherford Discovery Fellowship. And really this leads to a whole holistic approach to research that is ecosystem focused rather than a single species focused. So the first part of this research, and this really was Nick Wiper's cry for help, he involved me, and this was part of the old core program, he involved me in trying to understand how the land use changes affects the growth and survival of this pathogen now that we've identified it. Now what they meant is they didn't know, MPI and Department of Conservation weren't sure about how this tracks through the landscape system. And we were all working with Waipoua Forest, so Te Roroa iwi. And for Waipoua Forest, for those that don't know, it's probably about our largest intact primary forest that still has primary growth forest, it still has the main trees, but around it has patches of Pinus radio plantations and pasture as well. So they were trying to figure out, because vectoring's a massive issue, how did the pigs and how did machinery and how did the people move the pathogen from one side of, say, pasture into coyote forest, and how does this impact on its potential to be a disease reservoir? So that was the first stage. And this research was carried out by Kai Lewis, an MSc student. And what he did is he exposed, we grew up the Phytophthora eclodicida in mycelium in America, so we had the mycelium mats, and we exposed them to soils from the different land use changes. And what Kai found was that in pasture, we showed an increase in sporangia, so that's the fruiting bodies that release the zoospores, and in the Pinus radio land use, we had an increase in oospores, so that's the survival spores that can hang around for years until the conditions are right. So the surrounding land use potential had shown that there was potential there for pathogen reservoir or disease reservoir, which means that when you track through systems, and this is what Te Roroa and a Department of Conservation were interested in terms of pest management, then we had to be extra careful in terms of hygiene between moving machinery and people between these land uses, and as well as controlling those vectors like pigs that go through pine forests and into kauri forests. So that raised interesting questions. It's like, what was driving the effects that we saw? And this is where Alexa came in to look at, is this a biological thing, or is it a chemistry thing, or is it a combination of both? And what she first looked at is what's the soil microbiome difference between Pinus radiata and kauri forest soils. And what she found was that you get a complete change in fungal community structure between the two. So there's basically two divisions shown on this very pretty diagram that looks pretty, but it can be very difficult to explain. But basically there's two divisions, and in the kauri soils, which is shown in the green here, you get a dominance of this Basidiomyca, I'm going to say that wrong, division of fungi, and in the pines you get an Ascomycota sort of dominance. We don't know what this really means in terms of function, but that's interesting. It may be obvious that you would get a change, that kind of fungal structure, when you shift from one land use to another. And we took a deeper dive into, okay, so what does that mean? So you've got, it's obvious that you might get these land use changes and changes of soil microbial communities, but what happens when you get diseased trees and forests? How does that change the microbial structure? And what does that mean for function? And when I say function, I mean, how does it relate to carbon storage? How does that relate to nitrogen cycling? How does that relate to phosphorus cycling? All these kind of ecosystem functions that drives primary production. And so her site was also around Waipua, and we managed to we were invited to sample around Tānamaka, one, to see if there was infected, two, to understand the soil microbial community around that significant tree. And so what we found is that, again, when you've got the presence of diseased trees, you get a complete shift in fungi communities. In the diseased trees, you see a lean towards saprophytic fungi, which kind of makes sense if you think about it. And you also get a shift in those carbon cycling genes. And what that means is that we see a lot in those diseased tree areas, we see a lot of genes associated with carbon degradation. So if you extrapolate out a little bit, you can think if we've got the presence of diseased trees and large amounts of them, we're shifting, not only we're shifting community structure, but we're shifting towards becoming from a carbon sink, which a lot of soils are, to a carbon source, carbon dioxide source. So that does have ramifications about how we manage carbon budgets in our native ecosystems. The nitrogen gene cycling story wasn't so clear, and neither was the bacterial study. Another part of this was looking for any potential that we could find, some sort of tool that would allow us to probe a little bit deeper into potential soil disease suppression and microbial antagonists from indigenous soil microbiomes. And with the aid of some of Te Roroa workers, they took us to some healthy forest sites where they believed this was disease-free, and we confirmed it was, but also this looked healthy to them. And by miracles, Alexa managed to isolate Australian penicillin and Australian Burkholderia. And I'll circle back to this and we'll talk about that. But that means there is potential there to come up with some sort of antimicrobial tool eventually that can help in this to fight kauri dieback. So just to recap on what I've kind of covered, the dieback disease does alter the structure and function of kauri soil microbiome, and we can identify potentially beneficial microbes associated with healthy kauri. And the kind of research does allow us to look at monitoring these long-term trends for impacts on functions such as carbon storage, phosphorus and nitrogen cycling, really critical in these forests, and we don't really understand the dynamics there. And I I guess we go on a little bit of a tangent, but it's still part of the bigger picture. So if you follow me, you realise that this research is a little bit fragmented, much like the landscape itself, but it does come together in a big picture, trust me. And in came Alana, and part of Alana's thesis was here to trial some of these antimicrobial compounds that could help in Coaty dieback. When we were working on Coaty dieback a few years ago, honestly, everyone sent you everything that was going to solve everything. And some of the stuff that they sent you to trial, you could have used in clean up crime scenes. It's that bad. So, yep, could have got rid of Coaty dieback. We got rid of everything else as well. So we went through this process and eliminated all these solutions, I'd like to call that, to narrowing down some new to the market antimicrobials that were used as antifungals, I always get that wrong, in nursery trays, but also 4 essential oils that have also antimicrobial properties, and that includes Kānuka and Mānuka. And what Lana did find is that those ones designed to be anti-mycetes or antifungal definitely did a better job than those essential oils. But the Kānuka story is interesting, and I go on because another part of the research, and I was involved with University of Victoria scientists and some iwi working out in Northland that included those around the Kaipara Harbour, was co-developing tools guided by Indigenous knowledge. Now, we had a suite of plants given to us to test, and one of them was Kānuka. We had a whole bunch I could talk about them. One was Poraporo, Mānuka, Kānuka, Kawakawa, all those sort of plants. But it was Kānuka itself where we identified 3 new flavonoids, and when we exposed the Rhizos Phytophthora agathodis to rhizosphere soil taken from Kānuka, it had very much an inhibitory effect. So there is something definitely there. Whether you take that and develop that chemical formulation and spray, or you just plant a whole lot of Kānuka out, that's something that we're working towards actually with my colleagues up there. But I mean, there are glimmers of hope that there's something out there that we can do. And I've talked a lot about what goes on below the ground. So here's my opportunity to talk a little bit what happens above ground, even though what's on the surface litter. So Lauren Waller came in, and this is partly funded by the National Science Challenge, Biological Heritage one, where we looked at the role of exotic plant species in these forests. And Lauren concentrated on the leaf litter composition. And we were thinking perhaps exotic plants, might pose more of a, asymptomatic host for disease reservoirs for Agathodicida than, say, the native species. But what we found is really there was no difference. Even though the exotic ones looked like they were sort of more delicious to Agathodicida, it ended up not actually mattering. What mattered was the isolates that came when we exposed it. So it was more an isolate thing rather than a plant thing. And so if you remember, I talked about the isolation of penicillin and Burkholderia. What did we do with that, or what did Alexa do with that? Instead of going around and trying to isolate like a needle in a haystack any potential microbe that could produce anti-phytophthora activities, what she did is took a little bit of a shortcut, but a smart one. And so with permission from Te Roroa, we sequenced the Burkholderia and penicillin isolates to look at those biosynthetic gene clusters. So those are the regions of genes that produce secondary metabolites known to have produced antimicrobial properties. What does this mean? Effectively, what does this mean when you're working the soil? So you have soil microbes, and like fungi and bacteria often release antimicrobial properties mostly against each other. And this, being able to just identify those gene regions in soils, we can cover quite more area than we normally do with isolation. So we're looking at specific regions of forests that are able to produce these disease suppressive properties. And these could be, because we're still not sure why some areas have the disease and some don't. And so this could be, we hypothesize this could be a reason for the inhibition of Agathodicida that we don't see in some forests. So we're going around and we are sampling a whole range of forests at the moment around Northland. One of them is Pukiri, so go across a range of forest and disturbance sites. We're hoping that understanding where these naturally occurring, we hypothesise the naturally occurring disease resilient soils are, will help with the restoration and management of coyote forest. So we do find these, we can ring fence them and try and understand how we can build upon that, like little sort of disease suppressive nodes that we can build out from them. And also taking one step further, identify those specific bioactive compounds those genes actually code for, and utilising that in the biocontrol of Aotearoa. And this is just not published yet, but there were two gene regions that stood out, and one of they both transcribed for I guess what we call kind of broad spectrum proteins that produce a range of biological needs, including antimicrobials such as antibiotics and sort of immune suppressant type of compounds that we're used to seeing. But one is a non-ribosomal peptide synthase, so that's the one on the, again, I won't point, but it's the one on that side there. And this changes weirdly with forests. It wasn't so clear on the other one. But so when you see a change in genes being expressed between forest types, that indicates there's something really cool going on, or something really tricky we can't explain. But anyway, it definitely makes us want to understand why this particular gene is expressed a lot more in these forests than others. And these genes are expressed by bacteria and fungi mostly. So again, it could point to a change in microbial shifts, but why is Puketi forest so special in terms of this region? So what are some of the outcomes there? We see that less disturbed, healthier coyote forests have higher diversity richness of that particular gene region, the non-ribosomal coding one. We want to move from that taxonomy-based methods to looking at more microbial functions, because at the end of the day, we're not sure it really matters who's there, but what we care about is the functions that they do. And we're looking at, by using that kind of biosynthetic gene cluster method, looking at alternative tools to screen for antimicrobial potentials of these soils and forests to protect against Aotearoa. And just, again, I digress because this happens in the background to me all the time. I get involved in outreach and education in coyote dieback, and that is working with a lot of schools up in Northland, and that's with a lot of my colleagues here. Some are artists, some are musicians, a lot of scientists, and we developed programs for these schools up in Northland that involve some of their pūrākau stories, and we merge them with science so they can appreciate, I guess, the context that we're coming at, and it makes them want to engage more with the kind of work we're doing and being their own kaitiaki, but they understand that from a science base. And that picture down the bottom there was taken before Wainau and Aotearoa with 200 more students. But that's kind of a fun thing on the side, and we've also hosted students down here at the whare from Ngāpuhi as well. And it's when you work in conservation space, you need the buy-in from the community, otherwise it just doesn't work. So I'm just going to talk about the new research opportunities here, relinking those keystone species to promote soil health, and democratising genomic tools for real-time surveillance. This is kind of the heart of my Rutherford Discovery Fellowship, and that is, from everything I've talked about, is looking at those changes in structural communities, the presence of diseases, and the land use, the impact that land use has, and the change in sort of the functions that we see. When how have these, if you look back in time, I've talked about land clearance, fragmentation, and the introduction of unwanted organisms. How have these ecological transformations impacted on that soil microbial community and their functional responses, for their ability to sequester carbon? And when we want to even go back even further than land logging and land use clearance, New Zealand had birds, had lots and lots and lots of birds. And this is from unpublished PhD thesis in Auckland. And this is part of a New Zealand Geographic article that we contributed to. Now, if you think back, prior to we got fertiliser coming in to do agriculture, the only fertiliser that came to our systems were from birds, right? And you see the range, the extent of birds had. Now they're gone, right? Apart from a few offshore islands and in pockets on the west coast and some in the Hauraki Gulf, which is where we're going to talk to next. they would have been the only source of phosphorus, apart from some windblown stuff, that we had in our system. Now that's gone. What are our forests actually going on? If they run out of juice? And what does that mean in terms of soil health and function? Introducing Sid. So Sid has begun his journey with us in his PhD here, and we're going to work on understanding what this chronosequence looks like on Aotearoa, so Great Barrier. It's part of the Hauraki Gulf, and it has all these elements in it that we can hopefully try and understand how the seabird population has contributed to the soil health and resilience. And if there's a case that we have resilience that's produced by the presence of seabirds and their guano, what does that mean in terms of recreating those resilient links? Is that another case for, instead of focusing on, just preserving a species, we look at bringing back those keystone species and ecosystems that are under threat to actually kickstart bringing back resilience into the system. And I come back to one of the examples I come back to is a friend of mine who works in Canada. So they looked at returning the bison to the plains, and they noticed when they returned to bison, which is a keystone species as well, they noticed the return of invertebrates, plant species, all sorts of things, because these things had such an impact. So this is an unusual concept, but it's one that we've drawn together through conversations with other researchers, but also with kaumātua that have observed the changes in terms of plant species as well, where the birds have disappeared, plant species have gone too. And this is just, we're going to go there next month, it'll be great. We've picked out some sites, as long as Doc play the game, we'll be all good. So what Sid's going to look at is, first of all, doing some metagenomic work, understanding the communities, who's there, and the different gradients of ecological disturbance, and taking some soils and from the birds, when we're allowed to get near them, those ones that are influenced strongly by seabirds, because there still is very much strong seabird colonies on these islands, and exposing them to phytophthora agathis and see what happens, and see if we can actually go back in time and recreate those links. Should be exciting. I'm excited. And this, lastly, this is a program that I'm working with still with Genomes Aotearoa. We haven't quite landed there yet, but it's really about democratizing those genetic tools. And there's really good reasons why you do this. One, it saves time, it saves money. At the moment, there's 2 labs that are only able to do diagnostics on ecothodicida, costs a lot of money. A lot of the sites that we go to are remote, it takes days to get there. And so lugging samples out of the forest and waiting weeks to get results isn't particularly good when you want to manage something. But the trick is getting DNA out of the soil on site without a PCR and the centrifuges and everything else that you need to cut with you. So we're looking at creating that pipeline, that diagnostic pipeline, doing it on site and field, which will open up a lot of avenues for those remote communities who want to do their own diagnostics and lead to maybe other metagenomic applications such as conservation. We're trying to understand what species you have, presence of pests, all sorts of things, but just good all round. And it gives them the ability to manage their own data. So data sovereignty is a hot topic, as well as repatriation of soils. That's not becomes an issue. So that is on the cards as of next year, I hope. That is me. And I had a slide and I had everyone that I wanted to thank, and it looked like hundreds of thousands. But there is a lot of people I wish to thank. Lots of people from MPI, from Department of Conservation, from Plant Food, from Scion, from Lincoln University, from Otago University, from Te Roroa, Ngāpui, Ngāti Hini, Ngāti Wai, Te Kawaramaki, our Auckland Council, and Northland Council, and a lot of Forest and Bird also, and a lot of other sort of NGOs that were involved in piecing together all this research that hopefully I've delivered to you in some sort of coherent package. But thank you for your time.
That was fascinating.
We,
our own family property in Wellsford, which is close to the
Cody, do you need to test it?
We have it is 400 hectares, and it was probably would have been covered 60% at one point. Well, currently there was one left. There you go. and it's in a little remnant. I still think it's there. Hasn't
Hope it's there.
it is
should
like
check.
that. So it is something that has happened, and certainly every time I travel to the north and do the trip around and come down past Aotearoa and come past Waipua Forest, and you see it, you see those skeletons there. It does really impress upon you the nature of the problem that we are actually addressing here. So a fascinating presentation and plenty of time for questions from you. So
Thanks.
open for questions. If you would just state your name at the start, Phil. So state your name so everyone knows where you.
Hi, thank you. First of all, Amanda, the issue of when curry dieback hit our shores seems to have historically been a bit confused, different views, etc. Some suggesting it's an introduction from this century, others saying it predates Maori settlement. So has there been a conclusion as to when it is? And isn't that an important, I mean, understanding the pathway would be quite a useful way of understanding the future risks?
Yeah, I mean, the jury's still out on that. And there was also, to add to that, there was also the discussion that's been here forever, since time immemorial, it's native. We don't think so. And the reason why, and I've talked to Bevan Ware about this, we don't think so, just because of the impact it has. But also when you look at the molecular, when you sequence them, the isolates, there's so many different isolates, it just sort of points to multiple incursions rather than one introduction, I say 300 years ago, and it's just spread out. So it's quite rapid multiple incursions. And I know Pete Lockhart and Rich Wentworth looked at molecular dating, but the model they use is quite different from the Dissident model. And so there's a lot of inherent error in that. So it could be 100 years, it could be 300 years. It's really hard to pinpoint. But even talking to them, we still come towards about 100 years, maybe the maximum, which kind of fits with the New Zealand forestry anecdotal evidence when talking to nursery people, and that when you go through the historic archives, that's where they think, and also the visual symptoms on the trees. Yeah.
Peter Armand, the Soil Science Department here. Amanda, there's lots of preserved kauri, obviously. Just relating to that question, going back through the last glacial period, there's 30,000, 40,000 year old kauri in good condition. Would there likely be any remnants of any previous infections that would preserve? Is it possible spores get preserved that long?
No, it's a good question, and people have looked at that. There doesn't seem, and they haven't, because the nature of swamp kauri, and when they get it, they kind of, it's like gold, they kind of hide it, so locations are not known for that reason, and so it's really hard to get specimens. But the ones they've got hold of, I haven't seen any indication of the kind of infection through the xylem, which is what you see. So
I have a colleague at NIWA has a huge collection of kauri swamp wood for dendrochronology and radiocarbon dating. So that archive is there if anyone wanted to.
I think it's been explored. Look, trust me, when this kind of exploded, it was just like lolly scramble. Everyone went in every different direction looking at different things. But thanks. Is that all?
Well, thank you very much for that. Very interesting presentation, Amanda. I'm interested in the seabirds bringing nutrients on shore in relation to the introduced farmland of pasture and pines, because not only they've changed the vegetation, they've also changed the biogeochemistry of those systems by bringing a whole load of extra nutrients on. Have you looked at that at all in terms of not just the species on those changed land types, but also the different processes that are going on and whether those are impacting?
I'm sure there is. It's funny, the chemistry didn't really reflect any particular differences that we saw, because chemistry is really stable and you have to put a lot in to see changes. So biology in terms of this is the best indication of anything happening. I mean, there is likely some spillover effects that you might see in adjacent land uses, but because we didn't see a clear signal in the chemistry, we didn't follow it up. It's not to say it's not there, so we didn't see it, the questions we were asking.
My question. Amanda, you're showing that the soil microbial diversity is different with diseased trees. And I was wondering, is it because the tree is diseased, or is the tree diseased because the soil has changed? Do you know what came first?
Yeah, no, I know. We asked that too. We can't answer that actually because it could be either or. And so those are the sort of follow-on experiments that you need to. It's chicken and egg. It's like, is it there because it's there, or is it something else? We can't definitively answer that. We suspect that you get a shift because the trees are diseased, rather than, you know, you've got a change in microbial community and that causes it to be less resistant to Phytophthora invading. Yeah.
James, perhaps before you ask your question, you were instrumental in getting the kauri tree grow as part of the 125.
Yeah, we had to establish a relic so that there'll be some living kauri in the country.
So I'm sure you're aware that we did, as part of the 125th year, it was 125, wasn't it? Yeah, celebration for the university. There was the kauri grow planted here on campus, just to the side of the Forbes building, apart from the one that's got a sort of split head that's going off in two directions. It appears to be growing well at this point. So thank you, Dick, for your contribution to that. But I'm sure you have a question.
Yeah, Amanda, there's been a lot of publicity about closing walking tracks and so on. Is there any real evidence that we're protecting the tree by stopping people walking, given that there's a lot of wild pigs, possums, rats, all other mammals doing their thing? I'm not speaking in favour of Colus bolus tracks everywhere, but is there any evidence?
It depends what forest you're talking about. If you're talking about the Waitakere, which is Auckland's playground, then keeping people out here, it does help, because when they do the surveillance of, you know, where Aotearoa is, you just have to go 50 metres off the track. It's all there, and the more into the forest you go, the less of it you get. So it is being spread by humans in that forest. When you go up to Northland, they're infested with pigs. It's pigs. So those are the main vectors rather than people themselves. So in terms of those forests that are regularly accessed by recreational users, it does help. And they put a lot of effort.
Would be good in the pig areas.
1080, yeah. They don't use 1080. They do use traps. 1080 is ineffective with pigs, really. They have to use hunters and traps, and you can't outshoot that problem. Trust me, we've looked at the numbers, because they breed so fast, and they're so destructive.
Thanks, Amanda. That was amazing. I did try and follow it all.
Sorry. I gave a lot.
Brilliant. It was brilliant. What an amazing piece of work. Two questions. One is, how come they're growing there if it's a subtropical species? And maybe we should be planting more. And the second one, how fast is this spreading? I mean, there's been keeping a watch on it. And are we likely to lose our kauri forest before we get on top of it?
You're going to lose parts for sure. I mean, once they're in a tree, the trees, I think, the mature trees, once they get it, it takes many years for them to die. They do die eventually, I think. No one knows how fast it's spreading, because the surveillance can't keep up, because it's so expensive. And we also get into arguments about people not wanting to know, you know, and so we, you know, don't want to know if it's there or not, or we get into the data sovereignty issues, or we get into repatriation issues, and then we don't have the budget. And so there's all of that. Now, this is an interesting question, because I was always led to believe that it is a subtropical species, and of course it thrives in warmer climates where it comes from, but it can survive here. And I think if you're looking at conservation long term, then you would want to have a refugia of kauri in the South Island where Aotearoa can't survive. Yeah.
Oh.
It's also
It's a subtropical Phytophthora. Yeah, we suspect it's closest to one in Australia or New Caledonia, if you look at the phylogeny of it. Yeah.
With climate change as well.
And with climate change. So it might come to Nelson or something like that. But yeah, I mean, if I could do anything, I would be create a refugia in a place where the disease doesn't exist. Yeah. Starting with your trees, too.
We'd have to do some agronomic work to see what sort of fertilizer it likes.
They said one of them.
We'll get some birds.
Sure. Sharon Jacobs, Agri-Surgeon, Genomes Aotearoa. I think you sort of answered my question, but I'd like you to elaborate a little bit more on it. You seem to suggest that it could have come in with related species that were introduced into the country. So have you gone back to the origin of where those trees were brought from and looked at what Phytophthora is there and what do we have? And is the local population, if I can say it that way, is there any form of resistance against or any form of fight against those local strains of Phytophthora?
I haven't, but people were interested in doing it. I don't know what happened. That was Pete Lockhart's kind of research interest, I think, is looking at potential origins of it. And I think funding reasons that didn't go anywhere when it came to prioritisation. The government put out $20 million that kind of got cut off more in favour of pool development and surveillance. But yeah, I mean, it could be picked up again. It's just someone would need to fund something so specific, a hunt for Phytophthora origins. Yeah.
If you look at Phytophthora infestans, which is the causal agent of late blight in potatoes, it's hugely variable. So I'm just wondering to what extent this beast, it's not a fungus, this mycete has that same variability to it.
Probably a lot. I mean, we just only know what we managed to sequence, and I'm sure there's a whole bunch of things out there we don't know about. You know, so you could fill in the tree. It's just a matter of finding someone to fund it and do it. Yeah. I guess I can't answer, whether that should be fundable or not. I just haven't done it. Yeah.
Amanda, working with Maori communities, it's been a key part of your work. Can you just talk a bit about it?
Yeah, sure. I guess I was introduced to those already working in that space, and when you work with Māori communities and other isolated communities who often are underfunded and sit in the margin and have a million things going on, you got to remember where you sit in their priority list. And so when you work with communities in Northland, some of their priority individuals are tree health and forest health, but it's more along the lines of employment, drugs, youth suicide, schooling and things like that. So you're asking for people's time and you've just got to fit in with them. And it's about developing relationships. And so it's a long-term thing, and often that doesn't fit in with the funding cycles. And so when you do develop a relationship, you've got to be in it for the long term, whether you get funded or not. You've just got to continue developing your networks and relationships and up there. And I guess that's what I've done. And they know me as someone who is going to be there when they need it, and it's going to do exactly what I say. I'm not going to do something else. So, and that's kind of reputation and that you build upon, so you're able to continue to work with these communities and other communities, because your reputation matters in this space, I think. Does that answer it?
Yeah, beautiful.
Okay.
Any final questions?
Grant, I think this is more a question for you than for Amanda. I'm always trying to think out of the box and stuff like that. We have a beautiful arboretum. Would there be space for some kauri in our arboretum? Just a question.
Probably.
Yeah.
Yeah. We started that. It's been 125 years.
Yeah.
Tracy.
Tracy Nelson, looking interested. Just further on the Maori side of things, and with the current debate around genetic modification, if a tool became available that involved GM, how receptive would that be for Maori?
It's interesting because we've just got it funded on an MBA that looks at development of gene drives and how that is socially contested. And it comes down to specific groups and their needs. And so you could go out and get a wide array of opinions on this, but what matters is the people that actually have the problem. And I think there's some groups that are anti this and anti a whole bunch of other things, and that's just that. So you work with people that want to be engaged in this space, and there are iwi that are interested in this. So you just work with them. What I'm saying is the opinions vary, and you just work with the ones that want to engage with you, and you don't try and convince the others otherwise. Yeah. You pick your battles and you just go with those you want to work with. Yeah.
Well, thank you, Amanda, for a fascinating presentation. And
Oh, thanks, Brett. It's Christmas.
yeah, and thank you very much for this opportunity to join for out in the foyer.